in human and web server categories. In CASP, we just went for human prediction. And in this type of category, we use just RNAComposer, that is our system we developed some time ago, and natural intelligence. So here is how we go in prediction. So we start from looking for any information about the target we can find in all available resources. So some databases, literature. Then we run RNAComposer. It predicts the 3D structure from sequence or the secondary structure. So when we obtain the results from RNAComposer, these are our initial models, and we analyze them. And sometimes they're good enough, so we just submit these models to CASP for RNA puzzles. And if there is anything wrong with the structure, we just try to remodel it. So sometimes it happens that some fragments are incorrectly predicted, and then we try to model such fragments in semi-automated or manual way, and we provide them again to RNAComposer, and we run RNAComposer again with initial model and own provided fragments which are wrong. So we run RNAComposer in a kind of homology modeling way. And then when we obtain the results again, we collect all the models from the initial modeling, from the second round of modeling. We analyze them, sort by energy, filter some wrong models with entanglements, we cluster them, and we select representatives of the clusters. Okay, so no deep learning. From the deep things we use is just deep sleeping. Okay, so in CASP15, we submitted 61 human models, and I will just show you 2 examples. So the first one is our models for these 2 targets. So there were similar targets of the length of 118 nucleotides. These are RNAs that act as protein sponges. So they are quite similar. One of them is in a complex with 2 protein dimers, and the other one with 3 protein dimers. So what we did first was scanning the resources. We found some consensus structure in alpha. We also found a paper from Nature from 2014 with 2 structures there, 2 MF0 and 2 MF1, and we decided to rely on these structures as templates in our predictions. The important thing we found there were these hairpins that you can see. So this is the template of this conformer L to MF0. This is some other structure we also found in the databases. So what is important there are these hairpins with protein binding motifs GGA. So we knew that our model should have these hairpins, and that pairs of these hairpins just hold proteins. So what we did first, we ran RNAComposer just from the sequence, then from the secondary structure that we found in alpha. And we tried to compare what we obtained with a template with this structure. And what we found there in this initial model was that the domains that interact with proteins were incorrect and had incorrect spatial arrangement. So we found it a challenge for us, so how to make a model to have these domains correctly positioned in space. Okay, so what we did next, we tried to find as many hairpins, 3D structures of hairpins in experimental RNA structures, and to match these hairpins in our template model, sorry, template secondary structure, and we were aligning these hairpins onto the template, this MF0 from the Nature paper, to find out the best structures of these hairpins oriented in the 3D in such a way that they would fit protein dimers. So from these trials, we selected several hairpin structures. Then we added single-stranded elements, so these linkers in between them. And we put this all together into RNAComposer. We run RNAComposer to assemble the fragments and minimize the total energy. And we obtained this part of the model without the terminator. So there was a terminator in addition. So we also modeled it separately, and we put these two fragments together to RNAComposer again, and we made it assemble both fragments. So as you could see here, we tried, RNAComposer tried different positions of the terminator with respect to the rest of the structure. Okay, and here's what went wrong and what was good. So from the secondary structure, we assumed that these two hairpins will hold protein dimer, and these two will hold the other protein dimer. So in this case, we were right. In this case, we were wrong. So as you can see here, this the secondary structure of the target, and you can see that this let's say blue arrow shows the same pair of hairpins as in our model, but this one is different, and there is another one in one of the targets, this 1190 that interacts with 3 protein dimers. So in this case, in these cases, we are wrong. So anyway, we obtained the models in all our models. Positioning of these two hairpins was okay. Also, the terminator was correctly oriented. These hairpins were not so good. So global RMSD of the best model was around 18.7 angstroms, which I considered okay. We failed, but as we compared to the others, it appeared that in general the target was difficult. So the best RMSD, as Eric showed you, was around 16 angstrom. Okay, and this is the second target. So this is this artificial RNA. We have 2 target structures here. So the young one and the mature one, and they differ just by this red bar here. So this is 720 nucleotides in the structure, and this is the largest structure we have ever predicted. But apparently it was not so difficult to predict it, because it was already the 4th synthetic target in CASP that we predicted. In general, we also never before were predicting the synthetic structures like this. But since this was the 4th one, we already learned from the previous 3 examples, and 3 examples appears enough to learn if you just have natural intelligence. So from the literature, we knew that the structure was RNA origami, and we found it easy because in RNA origami you just have a lot of recurring fragments of the same type, and what you can see here is the secondary structure of this target. So you can see there are just hairpins and 4-way junctions, and that's all. Okay, so what we did here, we run RNAComposer again, and RNAComposer has several secondary structure prediction algorithms incorporated. So first we analyzed the secondary structures that were predicted within this RNAComposer pipeline. And we found, here are the examples of the secondary structures. What we found there were these 2, let's say, segments, quite similar as predicted by all the algorithms. But when we took a closer look at these structures, we found out that there should be kissing loops there between these apical loops. So we modified the secondary structure by adding kissing loop interactions. So this is these green lines you can see here. And what we did next, we just put this modified secondary structure into RNAComposer again. So it could use, the system could use this type of secondary structure to build the model. Okay, and next we just scanned the databases. Here we used our database, RNA-Frabase, for 3D fragments of the elements, basic elements that are in this synthetic RNA. So these were four-way junctions and kissing loops. In case of four-way junctions, there were two families of these junctions, H family and CH family. So we just decided to use this one because it's kind of simpler and fits our vision of the structure. Okay, so basically what RNAComposer did was it just took one structure for a four-way junction from this template structure and one structure of kissing loop from this RNA, and it used it to assemble the structure. So here you see how RNAComposer works. So it just takes the 3D segments and assembles them by adding one by one. And here we have 33 loops and 32 helices, some common base pairs in between these elements, and one element joining these two segments. Okay, at the very end, RNAComposer runs minimization protocols. So we use Siana to minimize the model in torsion angle space. What it did here to the models was it made the kissing loops to form properly, and since some of the models included entangled elements, some of the entanglements were removed during this step. Then there was the second step of minimization in which we use explore. So in this step, minimization just improved the geometry, so some bond lengths, angles, and clash scores. Sometimes the problem with such big structures is that minimization does not converge. So of course, what we had was a pool of models, of 3D models, and we simply removed models for which the minimization did not finish here. Okay, and these are the results. So finally, we obtained several models. All of them had the correct secondary structure. All kissing loops make clasps between the segments. And we found out that our models are more similar to mature conformer. So the best one was 9.5 angstroms. As compared to the young model, we obtained around 20 angstroms. We also analyzed the models that were not submitted to CASP, models generated for these targets. So we found out that we had some models that were more similar to the young form, but we just did not consider them. We decided that they are not so good, so we were submitting mature structures. Okay, and this is the team behind. So Maciej Janczak, Mariusz Popenda, Joanna Sarzyńska, Tomek Grzok, and myself. Thank you very much.
Thank you so much, Marta, especially analyzing this complicated case with the protein. One quick question? John.
So what is it about natural intelligence that's not codable?
Once more, can you repeat? Natural intelligence is?
So, I mean, I appreciate you making an emphasis on you're using natural intelligence, but
Yes.
there's some kind of process there. And I think we're all struggling to relate that to machine intelligence. Can you envisage coding in some way what you mean by natural intelligence?
What I mean here is, of course, what we do with the information we take from the literature, yes, and some, of course, some knowledge we already have. So as I showed here, yes, when we looked, for example, and the secondary structures, yes, we knew that there should be some kissing loops there, okay, and this just comes from this knowledge just comes from the experience from working with many models before, yeah.
So maybe one of the large language models working on the relevant literature could capture this, or you think that's?
Yes, I think it's possible. It's possible.
Apparently, you said you only need 3 cases for a human to learn it, right? You said
Yes.
you had 3 examples and then you knew the answer. So
Yeah.
it's better than my machine. So, yeah. Okay, our last talk. Thank you very much, Marta. Let's
Thank
thank
you.
again.